Okay, well, welcome everybody to Liverpool Skeptics in self-isolation. I hope you can hear me. I hope everyone's well. If you can hear me, if it's all working, feel free to say hello in the chat that we've got. Normally wouldn't allow chat during the middle of our live talks, but these are exceptional circumstances, as I'm sure you all have noticed. So the event this evening is put on by the Merseyside Skeptics Society. We are a non-profit organisation dedicated to the promotion of scientific skepticism, critical thinking, compassionate skepticism, science, and staying the hell indoors. What I like about this is that not even the end of civilization can prevent us from having our regular get-together. So that's a nice thing. Whenever we talk about how civilization would end and the apocalypse had come, who'd have thought that things would survive the apocalypse would actually be cockroaches, Twinkies, and the Labour leadership race, which still hasn't finished, despite civilization finishing all around us. But we're going to ignore that for the time being. We're going to have our talk from our guest speaker. This is something a little bit new, so we're trying out a new way of doing things. So you'll spot a few things are going on here. One of which is the chat. We're also saying that if you do have questions, you will occasionally and semi-regularly see a little link appear in the chat to a website called Slido, which Alice, who's going to be modding for the evening, will put that link in the chat. This is a website you can go to, pop in a code and ask any questions there, and you can actually go and upvote and downvote those codes, those questions, so we can see which questions people want asked, and we can use that during the Q&A. And you can see that Alice has put that in the chat there. Alice is going to be keeping an eye on the chat for the evening, and we do ask you to keep the chat respectful. We obviously the talk going to be going on. Alice isn't afraid to put you on the naughty step if she decides it is necessary. I'm your host for the evening. I'm Michael Marshall, and to get into the spirit, I thought I'd come along tonight with coronavirus. I thought I'd make that effort to really get into the spirit of things, and so I'll be popping up occasionally throughout the course of the evening, looking increasingly haggard. You'll see my face looking increasingly haggard over the course of the evening, a bit like the countdown timer on the TV show Nightmare. Just every now and again, I'll turn up looking a little bit closer to death. So bear that in mind, and please do refrain from telling me to take a sidestep to the left. Obviously, we'd normally be able to see the audience. We can't do that right now. And obviously, as a fantastic audience, you'd all, at the end of the talk, give a massive round of applause to our speaker. And you are still welcome to do that. But if you wait till about 8 o'clock, you might find that your neighbours even join in with that round of applause. And we can have a really good old fun show of things. So that's enough from me for now, because I can't tell you about any upcoming events that we've got going on, because the event has happened and we're all dealing with it. So I'm going to hand you over to our speaker for this evening. The topic, I think, needs no introduction. Look vaguely around you. But our speaker is Liam Brierley. He's an epidemiologist and research fellow at the University of Liverpool and a media ambassador for the Royal Statistical Society. His research interests revolve around using data to understand why viruses jump between host species and to predict future outbreaks of infectious diseases. And here to tell us what is going on with this coronavirus COVID-19 stuff, please welcome your speaker for the evening, Liam Brierley.
Happy also if people want to put in some clap emojis in the chat there. So yeah, as Marsh said, I'm Liam Brierley. I'm a research fellow at the University of Liverpool. I'm an epidemiologist interested in the ecology, the evolution, and the genetics of emerging virus outbreaks. So I'm obviously very interested in our current pandemic. And tonight I'm going to talk a bit about where it came from, but also the infodemic. Now my Twitter handle is there, it's L_Brierley. If you can't think of a question right now, but you think of one later, I'm totally happy to field questions through Twitter after this evening's session. So in the next sort of 20-25 minutes, what I'm going to do is I'm going to put the emergence and the outbreak of COVID-19 into the context of what's come before and what to expect in future. I'm going to review evidence for the origins of the novel coronavirus in a nutshell. And I'll also talk about how some flawed research has led to the spread of misinformation about the epidemic's origin. Now, I also think it's really useful for me to explicitly say here now what I am not going to do as part of this talk. So I'm not going to be discussing transmission and spread in this country or anywhere else, because what we know epidemiologically changes so rapidly, I can barely keep up. This is also not a talk about infection control or risks or symptoms. The latter because simply I'm not a clinician, and the former is because I can't really say anything that hasn't already been said. So it probably doesn't actually need an introduction, as Marsh says, but to reintroduce our current topic, I wanted to start with maybe a quick lesson in names. And that's not just because I take unreasonable joy in needless pedantry, but it's because there's been a lot of confusion about the names of the virus. So we talk about coronavirus, but the word coronavirus really just refers to any of the viruses within the family group Coronaviridae. So this includes other epidemic coronaviruses, but it also includes some coronaviruses that cause a common cold, and it also includes many that don't actually infect humans. Now, coronaviruses is the plural of coronavirus, and the new virus that emerged in Wuhan in December is properly named SARS-CoV-2, whereas the disease caused by this new virus is named COVID-19. So if you're asking, will I catch coronavirus? It's a little bit like asking, do you own mammal, if you're trying to figure out if somebody's got a pet cat or a dog. And why are these names so different? Well, it's because viruses are named by the International Committee for the Taxonomy of Viruses, and diseases are named by the WHO, and they each operate by their own kind of different rules. And some have argued this doesn't really make sense, but for now, this is what we have to work with. And if we look at the current situation in terms of global case estimates, as of today, it looks like this. There's about 487,000 cases of COVID-19 worldwide, and these are just the confirmed cases. And these are in 175 countries. And we are still in an exponential rise of cases at the moment. So where do new viruses like SARS-CoV-2 actually come from? Well, the majority of human viruses, about 60-70%, are what we call zoonotic. And this means that they can be transmitted from non-human animals to humans. It's often animals that we have a lot of contact with, like domestic animals, or it's animals that we're closely related to, like primates, but it also includes many other wild mammals and birds. And SARS-CoV-2 is certainly not going to be the last brand new human virus. It seems very likely that we're going to continue to see outbreaks of previously unknown viruses, because zoonotic transmission is constantly happening, such that we recognize about 2 new human viruses each year on average. And that's been surprisingly constant since the 1940s, 1950s. Now, our most recent estimate was that there are 214 human virus species. So to move on to the epidemic coronaviruses, many people likely remember that SARS coronavirus emerged in China in 2003, caused about 8,000 cases, had a case fatality ratio, or CFR, of about 10%. And then later, MERS coronavirus emerged in the Middle East in 2012. This has caused about 2,500 cases to date. It still occasionally causes infections. We see the odd small outbreak starting of MERS. The case fatality rate of MERS is higher at about 30 to 40%. Now, in this image, what I've done is I've scaled the area of these circles to be proportional to the numbers of cases and numbers of deaths. So if we move on to the novel SARS-CoV-2, it's caused many, many more cases than previous epidemic coronaviruses. It certainly seems like we're going to have over 500,000 cases by tomorrow at this rate of increase. Now, it's really important to note that these vary in risk. So while MERS-CoV is the most deadly, it's the one that you'd least want to have, it's the one with the most individual risk, SARS-CoV-2 has presented the biggest population risk. Even though the fatality rate is much lower, we've still seen an overall higher death count. This red circle is still bigger. Now, SARS-CoV originated in bats, but evidence has shown that it actually first transmitted to the masked palm civet, and this might be the cutest baby animal that I've ever seen in my entire life. From there, it transmitted to humans through contact in markets in Guangdong. The MERS-CoV originated in bats as well, and then it transmitted to camels. where evidence suggests it circulated in camels for a while before jumping to humans in Saudi Arabia. Now, the question now under investigation is, where did the current pandemic come from? How did SARS-CoV-2 reach humans? Well, to answer that question, we need a modern science known as bioinformatics. Now, what is bioinformatics? Now, your first guess might be one of the friends of a particular comic book Gaul, but bioinformatics is broadly defined as the science of processing, extracting, and understanding information from complex biological data. I'm going to narrow this down a little bit. I'm going to narrow this down here to mean the science of extracting biological relationships from complex genetic sequence data. Essentially, the genetic code of all organisms is made up of one long, big string of coded information, and viruses are no exception to that. And we can use that information to tell us things like evolutionary relationships. So, for example, here I've got some genetic code from 3 viruses, and it's a little bit like a visual sort of matching puzzle. If you just sort of skim your eyes over these sequences for a moment, hopefully you'll have already spotted that the top and middle virus are actually very similar, whereas the bottom is quite different. So this already tells us something quite important. It tells us that the top and middle are more closely related, and so one might have evolved from the other, or they might share a common ancestor, whereas the bottom virus looks quite unrelated. So we can apply these sort of principles and compare SARS-CoV-2 to viruses that we already know about in different hosts to try and find out which is most similar. And by doing that, we might get a clue as to where the virus has come from. Bioinformatic analyses have shown SARS-CoV-2 to have 96% genetic similarity to a bat coronavirus from a horseshoe bat like this one. So it seems very plausible this new virus originates in bats, just like the other epidemic coronaviruses. However, one of the areas that has the most differences between SARS-CoV-2 and this bat coronavirus is this part. It's a protein found on the outside of the virus, and instead, this looks very, very similar to the same part on the outside of a coronavirus that we found in a diseased Malayan pangolin. So here's this protein sequence comparison along the bottom there, pointing out the areas unique to SARS-CoV-2 and the pangolin virus, but not the bat virus. So it's exactly almost like that visual puzzle that I just showed before. Now, there has been second-hand reports that pangolins were kept around this seafood market where the outbreak started in Wuhan, but that still isn't really enough to say whether the virus has gone from bats to pangolins to humans without more investigation. There could yet be any number of other animal hosts involved. Now, for the original SARS outbreak, these investigations took basically the best part of a year. But we already knew this for SARS-CoV-2 in mid-February, because genetic sequencing of the virus was done super quickly within just 2 weeks. Now, kind of parallel to that, we've also seen a rise in the speed of open science and preprint sharing in the last couple of years. If you're not familiar with what a preprint is, ordinarily a scientific paper has to undergo this peer review from experts in the field before it's actually published and it becomes available to read. But many scientists now are choosing to make their work available before or during this review. And this has now become actually quite widely ingrained in academic culture. So here we've got the rate of preprint posting for a couple of previous outbreaks, including Ebola there in green in 2004, right through to just seasonal flu starting last year in yellow. And gradually, we've seen more and more use of preprints to share knowledge as an epidemic happens. I've just put a little notice there, by the way. All this data is open, and I've made the code to create all these plots available. So that's on my GitHub, which I can also link after this talk. So those are some previous epidemics. But if we look at the rate of preprints for SARS-CoV-2, we can see that research posted about the current epidemic started super early, just 22 days after the first reported case cluster. We had some preliminary research reports, and it's absolutely boomed since then to this kind of scale that we've never seen before. We've had almost 800 preprints in the first 3 months, and we've never had this kind of flood of information during an active epidemic before. Now, while the large number of researchers posting their research super early has led to some really important knowledge sharing, having all this unreviewed research openly available has been a bit of a double-edged sword in a way that perhaps we weren't actually prepared for. What I mean by that is that we've seen some unreviewed preprints get lots of attention that actually had unreliable findings. So one preprint that claimed to have found genetic similarity that indicated material from HIV had either naturally or artificially made its way into SARS-CoV-2. Now, the problem with this claim is that the segments of genetic code they looked at were very, very short. The largest segment was only about 0.08% of the genome. And that's so short, their information is really actually not specific to HIV. And the exact same code can be found in lots and lots of other organisms just by random chance, including that closely related bat coronavirus. So this actually doesn't give us any evidence whatsoever that HIV has contributed to SARS-CoV-2. And the best way I can describe it is I've got a tweet from biology teacher Jeff Witters here, because I can't think of a better sort of analogy for it. To have such a level of confidence from this to describe finding the same super short sequence in both HIV and SARS-CoV-2, to be describing that as uncanny is a bit like finding a short match of words in a rambling manifesto posted by a serial killer to a random line in a Harry Potter book, and then arresting J.K. Rowling on that basis alone. So this paper was highly criticised, and it was withdrawn 3 days later. Now, you might have also heard that snakes are the origin of SARS-CoV-2. Now, this came from an unreviewed analysis in the Journal of Medical Virology. And what these authors did was they essentially looked at the virus and the closely related bat coronavirus, and they looked at how often certain patterns appear within their genetic code, and they compared that to many types of animal. And this is the result. So essentially, the animals are the columns here, and whether it's red or green doesn't really matter. Just the more similar the colors are across these columns, the more similar the pattern usage. And they looked at this and they concluded snakes were the host of SARS-CoV-2. Now, I wonder if people can see what the problem is. Well, there's another bit of a clue in this, this tree at the top. But really, if you're looking at this figure, these patterns are actually showing SARS-CoV-2 to be really strongly similar to the bat coronavirus, certainly more similar than it is to snakes. So why would you conclude snakes are the host and not bats? Now, quite a few virologists came forward to discredit this based on the fact this particular flavor of analyses is actually never used to determine what kind of host the virus comes from. And quite a few epidemiologists, me included, came forward to discredit this based on the fact that humans share almost none of our RNA viruses with reptiles. That's not to say we won't ever see that in the future, but that alone should have put big question marks on this from the start. And in fact, several works came out after to formally repeat both of these preprints showing their flaws. So in both of these, it seems like biological plausibility has taken a bit of a backseat to immediacy in producing results. Bioinformatics is a super powerful tool, but at the core of it, that's all it is. It's just a tool. It has to be used in the right way. And that takes human critical thinking. And there's no advanced method, there's no AI or anything like that that is ever going to take away that step. The problems are caused by over-reliance on trusting outputs and methods without really dissecting what they're doing. Now, even though other scientists were actually quite quick to discredit these works, because they were immediately publicly available, their message had already gotten out there. And the WHO has quite appropriately referred to this, this uncontrolled spread of misinformation about COVID-19 as an infodemic. And it's tempting to hear that word infodemic and think that it only ever originates with social media or maybe print media. But considering the role of research, I would argue the infodemic is more systemic. If you look at Google Trends, which are measures that Google produce on popularity of certain search terms, the posting of these preprint articles was followed by an immediate rise in associated misinformed searches. So searches for HIV and coronavirus, they're in blue, and snake and coronavirus in green. Now, obviously, I can't be sure what the intent of these searches were. Some of this might have been fact-checking, for example. But what I'm trying to show is that these unsound findings did quite quickly filter through into public perceptions. And I've added this just last moment, actually, as the results from a particular survey are now available. This survey was done in late February, so basically just at the end of that previous graph of search terms. And one of the questions in this survey asked whether it's likely the new coronavirus is a manufactured bioweapon, which is how the results of this HIV preprint were interpreted in some media sources. And among 3,000 US and 3,000 UK respondents, just under a quarter said it was slightly likely or stronger in the US, and just under a fifth in the UK. And even that level of belief, I think, is actually quite staggering. Now, another piece of misinformation that I've added quite last minute is this. There was also a press article claiming that there was a link between the SARS-CoV-2 outbreak and bat consumption in soup. And this included a couple of what you might call shock images, despite these images not actually being taken in China, and despite there being no scientific basis for transmission via food consumption. Now, this was later revised to remove the link between soup and the outbreak, but immediately after publishing, we saw this spike in searches about coronavirus and soup. And I've included this just to show the level of misinformation related searches that came from this preprint research was basically just as high as the level of misinformation related searches that came from mainstream press media. So if unreviewed research is now seeding in actually a significant part of that infodemic, what can we actually do about it? One of these preprint platforms, one of these websites that hosts them, responded by putting up a site-wide disclaimer that reiterated all the research there is not peer-reviewed and it shouldn't be regarded as conclusive or used to guide clinical practice and so on. Leading scientists have also made public announcements and signed statements against conspiracy theories. But I think the core question is, if you consider now this huge amount of information that's out there in unreviewed research servers, whose responsibility is it to regulate all of this? Is it the responsibility of scientists, even though they already contribute quite a significant workload in doing the peer review in the first place? Is it the preprint servers, maybe through volunteer screening? And some of them do this, although nobody can be enough of an expert in all fields. Or is it even the end reader? Is it the responsibility of the public and journalists and the media to actually know the limits of what they're reading? And it's probably, the answer is probably quite boringly all three. Although I think this has shown that we do need some serious guidelines now as to how the general public should approach preprints. To people outside academia, this distinction between peer-reviewed papers and unreviewed research is probably not very clear. So this is something that I'm trying to write a skeleton of myself at the moment, to try and create some guidelines as to how the public can actually read preprint research in a more kind of balanced way and understand what the limits are. So I'm going to wrap up. Genetic evidence suggests that, like the other epidemic coronaviruses, SARS-CoV-2 originated in bats. But we need more work before we actually know whether other animals are involved for sure. we've had this gap between academics and the public that's been gradually narrowing as the use of these open access preprints has increased and increased. And as a potentially unexpected result, unreviewed research can be a source of misinformation during epidemics. And I would argue that we need to be better prepared to combat this, not just during epidemics, but also during future large global crises. We can do this through good practice and transparency about preprint limitations. So I might usually sort of finish up by including some sort of talk acknowledgements. Instead here, I wanted to acknowledge something that you can do by just making everybody aware. If you're not already aware of it, there is a platform that is trying to crowdsource volunteer time to help COVID-19 research. So whether you have practical skills, data analysis skills, coding skills, or even none of them. Now, I'm not affiliated with this officially, I should say, but there's more info if you are interested at crowdfightcovid19.org. Thank you for joining me tonight.
And I think from the comments, it all went very smoothly. So that's good. We've got the technical side of this. And I'm sure everyone will agree that that was an absolutely fantastic talk. Now, I think we're actually thinking about having a couple of minutes break for people to go and get themselves a drink, have a quick comfort break. And then we'll be back in maybe 5 minutes time for the Q&A. So if people would like to join us at 25 to 8, we can start the Q&A at that point. Okay, welcome back, everyone. I hope you all had a chance to get a drink and a comfort break and everything like that. And I hope you're all ready for the Q&A with our fantastic speaker, Liam Brierley. So I have the questions you've already submitted. Those are to sly.do, and there's a code that Alice will be able to drop into the chat in just a moment to remind you. But the first question we have is from an anonymous source who asked the question, we keep hearing that there could be a vaccine in 18 months. How likely is it that 18 months is anything like the actual timescale for a vaccine?
That's a very challenging question. I wish I had the answer to that. 18 months is actually kind of more realistic than some of the numbers that I've heard elsewhere. Vaccines are one of these things that people sort of instantly think of in response to an epidemic, but they do take a very, very long time to develop. Typically, the course of a vaccine takes years and years. Even fast tracking the Ebola vaccine, it still has been a matter of years before anything like that has worked. Although I sort of suspect that even though I don't really know, I'm not really involved with those kind of like pharmaceutical sort of developments. I can't say whether it's realistic, but what I can say with a bit of kind of sort of gut feeling more than anything else is that if there is a vaccine that's fast tracked for this, it will probably be the fastest vaccine we've ever had.
Okay. Excellent. Let's see the next question. Another anonymous question asks, should we be worried that the existence of two strains, the L type and the S type, means the virus is highly likely to mutate again soon, evading cures and vaccines?
Right. So this is referring to a particular analysis that looked like there were two particular types of the virus circulating at once. Actually, my understanding is that's kind of actually quite highly contested. And there's a lot of experts out there who feel like there isn't two separate strains going around, and it's just been kind of misinterpreted from the genetic data. It's kind of uncertain at the moment whether there are two strains, but the presence of two strains doesn't essentially mean that mutation is going to be a problem. So mutation is one of these things, again, that a lot of people ask questions about during epidemics, and we talk about it a lot. Most mutations for a virus aren't really going to drastically change it. And any mutations that we see in SARS-CoV-2 are probably not going to have that much impact. They're probably not going to be responsive to things like cures and vaccines. If anything does fail against the virus, it's usually because we actually didn't understand enough about the biology to begin with. It's not that the virus kind of evolved something in response. Viruses do evolve really quickly, but generally they don't evolve quickly enough over the single course of an epidemic to kind of change in that way.
Okay. So Cash from Nottingham asks, do these viruses have the same percentage death rates in the intermediary species, e.g. camels?
Oh, that's a really interesting question, because that's something that we are interested in looking at, not just in the intermediate species, but also in the originating species. So in the intermediary species, I don't really know. For camels, I suspect that they don't have anywhere near the death rates that they would in humans, because obviously we would have picked that up a lot sooner. If camels being a domestic animal, we'd kind of spot it if a huge herd of camels just suddenly died. What tends to happen is that the originating host that a virus comes from, especially if it's bats, they don't seem to suffer any particular pathology. And we still don't really understand why. For a while, there was this kind of idea that bats maybe have a low-powered immune system. And now there's been new research suggesting actually they have a super-powered immune system instead. So there's something kind of special about the biology of bats that means that they don't tend to get sick with the viruses that they have. For the intermediary hosts, even though in the case of the camels, I've said they didn't get sick because otherwise we would have seen it. Certainly that's not true for kind of other bat-borne viruses. So there's a virus called Hendra virus, which has been found in Australian bats, which goes from bats to horses to humans, and that's very fatal to the horses. The typical sort of pattern is if a host hasn't co-evolved with something, so it hasn't been infected with something for a very long time, then it's going to be a lot more prone to higher fatality rates. Whereas if we've co-evolved with it for a long time, then we kind of co-evolve defenses, we have strategies against it. Yeah, a good kind of comparison is if you take one of our common cold viruses, which rarely ever kill people, and it transmits to, say, a gorilla or a chimpanzee, it will almost always be fatal in those species, just because they haven't evolved with it like we have.
Oh, okay. Someone asked a question, is there a reason that the 3 listed viruses all started in bats?
Yeah. So we think bats are kind of the kind of originating host of pretty much all the coronaviruses. And that's true for another set of viruses as well. It's true for, say, the rhabdoviruses, which includes a lot of rabies-like viruses. They seem to be the hosts of more viruses than other kinds of animal. But again, we're not really sure why. It's probably a product of their immunology, but also their ecology. You know, some bats live in colony sizes of millions and millions of individuals. That could be quite a good environment for viruses to circulate. Some bats go into what we call torpor, and torpor is a little bit like hibernation, except it happens overnight, every single night. So certain bat species at night will shut down a lot of their bodily systems, like maybe the digestive system, perhaps also their immune system. So there's something about bats that makes them quite different to other kinds of animals, but it's still a bit of an open question.
Okay. We have another question here. How do new viruses actually come into existence? Is it mutations from one or more original viruses? And how much do they have to mutate before they officially become a new virus?
The real but boring answer to that question is some taxonomist decide somewhere. So, I mean, viruses, like anything else, really, are just sort of organisms, and we decide once they're different enough to be a new species. For viruses, it's that definition's a bit more slippery than other kinds of organism, because if we had 2 mammals, for example, we could say they're a different species if they can't breed and produce fertile offspring. But viruses don't reproduce sexually. They just make clones of themselves. So the point at which you say, yes, this is a new species, is actually really difficult to do. And a lot of people argue about this. And I know because I have to listen to a lot of arguments about this. But essentially, there's kind of expert groups that decide on what genetic basis something should be considered a new species. So like that example I gave of SARS-CoV-2 being 96% genetically similar to this bat coronavirus, actually they would be classed as the same species. But that might not be true for other kinds of viruses. So the exact definition depends on the type of virus you're talking about. But broadly, it is it's more of a human concept than a biological concept.
I see. It's not the viruses naming themselves or anything in that case, then.
No, that would make our lives easier as well.
So we have a question from a pair of Jameses. They asked, can we realistically prevent future species jumps of viruses?
I would argue yes. Yeah. And it actually wouldn't be that difficult. But the problem is, with this kind of, I suppose, more kind of prevention, it's difficult to convince policymakers to spend on it, because they can turn around and say, well, if we didn't spend on it for the last 10 years and we haven't had a pandemic, and now we've had one, I think it's going to become easier to convince a lot of policymakers. But, I mean, the fact is, if you weigh up the economic costs of a major disease outbreak, not even a pandemic, and weigh it up against the costs of putting in those interventions, going to, say, tropical communities where viruses are at most risk of jumping into people, looking at giving those people who work with animals or people who hunt wild animals for meat some kind of protective gear, some kind of education about risks of transmission. The costs that it would take to invest in that are a tiny, tiny fraction of what it costs us every single pandemic. I think it's been estimated it would cost about, I think it's about $8 billion US to go out there and understand the genetic code of and identify all mammal viruses. And it sounds like a lot of money, but the figure I always compare it to is it's basically the same amount that we spend on preventable alcohol and smoking related illnesses, and that's just in this country. So I think it's actually really, really achievable, but the difficulty is in getting the policy and getting the resources to do it.
I see. Okay, we had a question here from Andy, who asked, how are the experts predicting the peak of the curve? Is it methodologically?
Yeah, there's been all kinds of epidemiological models that have contributed to what our predictions of COVID-19 is going to do in the next couple of months. A lot of them contradict, and that's to be expected. No model is kind of perfect. They'll all have different initial conditions, they'll all have different assumptions. There's been actually quite a hot debate between what people are calling the Imperial model and the Oxford model, coming from different research groups. I'm not actually up enough on those to say which one I think is more realistic than the other. But with all of these things, I mean, we talk about the peak of the curve. Actually, there's going to be a huge range of uncertainty around that. And I think it's difficult to try and communicate that to people, because if you say, well, it could be anywhere between this and this, it's not helpful for policy. But at the same time, just giving one single figure isn't obviously realistic, because different groups will come up with different answers. So what was the original question again, Marsh?
It was how do they figure out the peak? How do they work out the what they think will be the peak?
Yes, okay, yes. So there's a lot of, I suspect it will have come from what we call SIR models. So they're essentially models that work on mathematical equations based on compartmental flow. So it's essentially like, what is the rate of people getting infected? What is the rate at which people recover? What is the rate at which people pass the infection on to other people? And if you have all these numbers, or you can estimate all these numbers, you can build up a system of mathematical equations that then describes that infection curve. There's a lot of different ways of doing that, though, as I say.
Okay, we have a question here from Warren, who asks, presumably some species are more likely to cross the species barrier to humans. Are bats a particularly bad vector, or do they just get a bad rap? Are bats pricks? Warren asks.
I would argue that they're not pricks, because I was just saying in the break, actually, bats do a lot of good. They do eat a lot of insects, they do control pests, and if it wasn't for bats, we wouldn't have tequila. So that's one thing. But bats are kind of as much of a risk, because they do tend to be quite virus rich, let's say. They do tend to carry a lot of different types of viruses that we understand. But actually, if you look at other kinds of animal, let's say we compare them to rodents, we actually have a higher contact rate with rodents. In certain parts of the tropics, rodents can live in kind of urbanized areas, they can live in farmland, whereas bats tend to be a bit more distant. So it's kind of a function of all these things. It's a function of what kind of biology does the animal have, what are our contact rates with it, are our contact rates increasing? And it's true that our contact rates with bats are increasing in certain parts of the world. But they're kind of, their bad rap, as it were, tends to come from the kinds of diseases that they carry. So, I mean, not just the coronaviruses, we think they're the original hosts of the Ebola viruses, of course, rabies, which is fatal in pretty much 100% of cases if not treated. Lots of other different kinds of kind of hemorrhagic fevers. So there's something about them that creates particularly nasty viruses, but it's not that we have more contact with them than other kinds of animal.
Okay. We had a question along a different line here from Matt from York, who asked, do you suggest any particular sources for keeping up with COVID developments? Something that's generally pretty reliable and accurate and to the current reputable science.
That's a good question. Yeah. So as much as there's been this explosion in research, there's been an explosion of different websites, different indexes, different news updates. I mean, I'll hold my hands up. I do struggle to keep on top of it myself, genuinely. There is one particular resource, and I think it's called the Coronavirus Handbook or something like that. I can make sure it's linked in the chat afterwards. It's basically a sort of ethical tech company decided to pull together all the resources into one place that were reliable. So whether it's for clinicians or whether it's for members of the public, it links to a lot of fact-checking services. It links to a lot of health information. So it's basically like an index page for what they think, and from having looked at it, what seem like the most reliable sources. So I can make sure we do link that afterwards.
Okay, brilliant. We had a question here from Andrew, who asked, do you think it might be helpful to give these viruses and diseases better names? He said it worked in tech. Everyone cared about the Spectre and the Millennium bug. Would that work for viruses?
It sort of would, but the problem is viruses are really tricky to name. So there are now rules, and rightfully so, about what we can and can't call viruses. So we can't name them after the animal that we first find them in, because of course there's no guarantee that that is the animal that it comes from. That's what basically happened with monkeypox. It's transmitted by rodents, not monkeys. We can't name them after sort of individuals or places, because that can create a lot of stigma around those places. We can't name them after certain institutes. Certainly, when viruses tended to be first discovered, they might name them after the particular institute where it was found. Again, that can cause kind of stigma. People can misunderstand, and there can be a lot of kind of public consequences. So the most kind of consistent way of naming new viruses is naming them after the disease they cause. And I think really COVID-19 is quite a unique situation in that we kind of found the virus and the disease at the same time. So previously, it would be that somebody turns up at a hospital with a brand new disease, because our sequencing wasn't as good, because our investigation wasn't as good back then. It would take us a while to work out what kind of virus it was caused by, and then it takes a while to work out it was new. And by then, we've got a good handle on what the disease is, so we can give it a proper name. With COVID-19, we kind of identified straight away it was a new coronavirus. So people started calling it nCoV. And we didn't really understand what the kind of disease was then. So that's why these names sort of diverged. A few people have suggested calling it, I think, HCOV-19 to at least be similar to COVID-19. But there's rules and regulations about these things. The most accurate way of naming new viruses is just naming them after essentially the sequence accession, which is basically like a little ID number that they get once their genetic sequence is identified. But then that creates problems because nobody wants to sort of talk about a virus that's a random string of numbers and letters. It's not a very catchy thing to do.
Yeah, and it's not cool. You know, it's not the kind of cool name you can use to discourage people.
Yeah, maybe we need to get entrepreneurs to name them or something like that.
So we had a question here. We had a couple of questions on this subject, actually, but this question sums it up, I think. In terms of the research effort, is the work being undertaken by Folding at Home, foldingathome.org? Is that of any good value in this area?
Folding at Home, I'm not familiar with. I'm guessing from the sounds of it, it's something like a kind of crowdsourced kind of thing where you can. I'm guessing it's about protein folding, something like that. So what I understand is that essentially you can project what is going to be the structure of a protein from the genetic sequence, but sometimes it doesn't quite work, and there's a lot of different configurations, and there's There's lots of, it's almost like a puzzle, and I think a few websites have actually, way before COVID-19, have tried to crowdsource this kind of thing by presenting it as, oh, it's a puzzle, can you solve how this protein folds? In terms of how it's used, it's a science that's so far away from what I do. I don't actually know, I'm afraid. I mean, if you've got nothing else to do while you're self-isolated, then sure, why not?
And I can see there's people in the comments saying they're using it right now, so maybe they will be able to explain that a bit more in the comments at a later point. But we had another question here from James and James. They said algal viruses serve a useful purpose in ecosystems. Do human mammal viruses have any plus sides, or could we eradicate them with absolutely no ill effects, do we think?
It's an interesting one because they're so different from bacteria in that respect. So while we have a lot of bacteria that we've co-evolved with, they do functions in our body, they help us digest things, we have like a normal skin bacteria, we have a normal gut bacteria and all this. Viruses, I don't think there's any evidence that any viruses are beneficial. There's a lot of them that don't do anything. There's many of them, they tend to be DNA viruses, so very different kinds of viruses to coronaviruses. But there's some that are basically infecting everyone. You sample someone's blood, it's almost guaranteed they've got these particular viruses. And they just seem to sort of sit there. They don't cause pathology. We don't really understand what they do. We don't really understand how they interact with us. There's, I guess the best analogy for it is there's none that do good for us, but there's a lot of passengers who are just along for the ride.
I see. Gotcha. We had a question here from Julie who asked, what's your guess of the real number of infected people in the UK or Europe or the world, since only people right now with severe symptoms are being tested? Do you have a feeling for what the numbers are more likely to be as a statistician?
I wish I did. I wish I did, because I haven't done any of these kind of models. I mean, certainly there's going to be a huge asymptomatic fraction. That's kind of what things have suggested already. That's the case for most viruses, to be honest. So I'm confident in saying the case numbers we have are a gross underestimate. Just how big they are is something that's really difficult to speculate about.
Okay. We have another question here from Matt from York. He asks, is there a move towards having some form of preliminary or other means of rapid quality control going on?
Yeah, there are a few actually. I mean, the kind of purported function of a lot of these sites where people load this unreviewed research is exactly for that to happen. It's for academics to kind of put their comments on it whilst it's undergoing formal peer review. There's some places that are actively trying to push people towards that now. So there's something called, I should get the name right, it's called something like Outbreak Pre-Review. And that's designed to basically allow people to request specific preprints that academics can then get flagged to them to say, oh, lots of people want a review of this, can you do it? But they're not actually really being used that much. So we've got this kind of challenge of how you actually create an incentive for people to do something, which if they're academics, they're certainly going to be doing anyway. And they struggle to fit that in as it is. So it's going to be tough to find ways of getting people to actually use these platforms designed to give this peer commentary. But it absolutely exists. It's just that it's just not being used to its full potential.
Well, as a follow-up question to that, Cash in Nottingham asks, even if there is this kind of quick review system, isn't that also going to be already too late, because the newspaper articles have already been written on the very preliminary study before the peer review kind of thing kicks in and points out the flaws?
Yeah, it can be. It can be. I mean, we would hope that journalists would actually wait a bit maybe and or at least try and get some kind of scientific advisory services on the preprints. I suspect that journalists haven't really done that because they don't really understand what the difference between a preprint is and published research. In some places, though, journalists absolutely do that. So in what I do with the Royal Statistical Society, basically we have a service, not so much for epidemics, but just for any kind of statistical query where journalists can come along and say, I want to report on this paper, I'm not really sure on the findings, or it looks like it's not reviewed. Can you just sort of help me understand what I can and can't say from it? So while the possibility is there for journalists to just take the information straight away, I think we can still encourage a lot of media sources to try and get some scientific consultation. Again, that's going to take volunteers, but I don't really see any other way that we could do it.
Okay. We have a question here from Warren. He asks, what's the worst or most dangerous piece of misinformation you've heard so far with regards to COVID-19?
That's a really good question.
Should I take a sip of water every 10 minutes while you're answering it?
Yeah. You could. I mean, something like that doesn't. It's not really dangerous, right? It just wastes people's time, and I love wasting people's time. But in terms of dangers, I don't know. Thankfully I've tried to sort of keep away from all of it. I mean, in terms of, I think that probably it's the sort of subtle things that actually create the most danger. So even though, yeah, people might be spreading misinformation that encourages people to do, let's say, risky behaviors when they should actually be trying to protect themselves, there's a lot of misinformation that goes around that just kind of seeds a general distrust in science. And I think that's the kind of most dangerous thing, because it's a slow burner and it affects everything else. And that's one of the other things that I kind of try and do in my other roles, is to try and sort of talk about the value of science, what it can and can't say. But if somebody has their faith in science undermined by something, then it's going to affect every decision they make, right? It's going to affect the decision of whether or not they get vaccinated or their child gets vaccinated, or it's going to affect their decisions about medication. It's going to affect their decisions about just pretty much everything on a daily basis. So even though it's not really that related to COVID-19, I wish I had a really kind of ridiculous example to give. I think that's my actual answer.
That's good. Okay. So I think we've got a couple more questions left. Matthias and Bonnie ask, you said that the virus evolves too slowly to change in a meaningful way, but because it's spreading so much, does COVID-19 have many more opportunities to essentially go bad?
Yeah, that's a good point. Yeah, so I mean, as an epidemic spreads more and there is more of a virus, then just, yeah, mathematically, we would expect that there is a chance of the virus changing its genetics to have an effect on what it does. But I think that doesn't really change so much that it's still very unlikely that we would see a drastic kind of jump in what we would call phenotype, which is essentially how the virus behaves or what it does, just based on evolution over an epidemic. So it's not, for example, that the novel coronavirus could suddenly evolve to get into a mosquito, because that's a huge biological change. What is within the realms of feasibility is that it might evolve or change a little bit more to perhaps slightly alter its pathology. It might become slightly more virulent, it might become slightly less virulent. We don't really know. And the chances of that kind of happening one way are just as likely the other way. And I think in these situations, it's also really important to say most kind of mutations to a virus will just stop it from working. But yeah, it's a kind of a lottery model in the end.
Okay, we have, I think, 2 questions left, because I'm going to skip over the people asking where can I get a pet pangolin and should we kill all the bats. I think we'll leave those questions for another time. We have the question here from Dave Jenkins in Coventry, who asks, how effective are the social distancing measures in enclosed spaces? He said, I work in a supermarket, and it's a very regularly asked question at work, is how effective these measures actually are in such an enclosed space.
I don't think we actually know in some ways. I think it's kind of the measures that we have are really just to be conservative. There hasn't been any particular study that I'm aware of that actually shows what the transmission rates are over certain distances, because that kind of study we can't ever actually set up. We can't set up an experimental chamber where we do this kind of thing with people for ethical reasons. So I think it's probably based on what we know about the transmission of other kind of respiratory coronaviruses and other respiratory viruses from other families. In short, it's something that is just a conservative measure. I don't think it's possible to actually sort of quantify, oh, let's say if you stand this far away from somebody, your risks are reduced by X%. If you stand this far away, they're reduced by X% or Y%. Doesn't really sort of work that way. And at the end of the day, it's quite difficult to actually ascribe where transmission came from in the first place. I mean, even if we had a kind of group of volunteers who told us how far they distanced themselves from everybody in their life who they know of, and see whether they get infected with the coronavirus or not, it's still wrong, I think, to attribute that to that social distancing when they could have got it from, they could have got it from the surface, or they could have got it from somewhere else, or they could have got it from somebody who they thought was infection free, because there's going to be a lot of asymptomatic people that spread the virus as well. So in short, again, it's challenging to know, especially at this stage.
Okay, I think we'll ask the last question here is from Dave the drummer, and he asks, can CRISPR-Cas9 assist in the development of a vaccine, do you think?
I would love to know. And I have no doubt that whoever the person is asking this probably knows more about it than me, because this is a very kind of biotech sort of thing. A lot of people are talking about CRISPR now as a way of addressing sort of new pathogens. They're looking at CRISPR as I think maybe somebody's even mentioned CRISPR in the context of sort of preventing aging. It's kind of, people talk about it like it's this biological panacea, and actually functionally it's something proteomics that, again, is so far from what I do. I wish I knew more about it, but I'm afraid there's not a lot I can tell you about that.
Okay, so I'll just, sorry about that disappearing. I've changed back from Alice, by the way, so thank you, Alice, for stepping in momentarily there. We'll just repeat the last question, which was, how often do the mortality rates significantly increase during population transmission?
Yes, okay, so for that to happen, for a virus to be spreading, it to be mid-epidemic, and for it to suddenly become more virulent, there isn't really any kind of documented evidence of that I can think of. There's no particularly good examples. What does come to mind is, what's documented is it's more likely for the inverse to happen, at least according to evolutionary theory. Because if a virus is spreading and it kills its host very quickly, then it can kill off its route of transmission. But if a virus evolves to become less virulent and it causes a milder disease, then the hosts are probably going to live longer. They're probably going to be transmitting that virus more efficiently. So the kind of classic evolutionary model is that viruses should become less virulent as they spread. And the kind of classic example of that is myxomatosis. So when it was introduced to rabbits in Australia, it was at first very, very deadly, and then very gradually it evolved to become less virulent. But again, that was on the scale of actually quite a few generations. That wasn't over the course of a single epidemic. Viruses really don't tend to change that fast, or if they appear to change that fast, then we've misunderstood something.
Okay, well, we'll call that the last question for the evening. So please, everyone at your respective computers, join me in thanking Liam for a fantastic, informative talk and a really fantastic Q&A. Liam, if people want to find out more about the work that you're doing or follow you online, where can they find you, and where would you sort of direct them to?
Twitter is probably the best place. I've got the very awkward Twitter handle of L_Brierley.
Gotcha. And we can put a link to your Twitter and a link to the various of the materials you want to share with us. We'll put those in the chat at the end. But thank you so much for making time to do this for us tonight. And thank you to everybody who came along to listen to this. This is the first attempt at putting out a Skeptics in the Pub online during the lockdown. This is Skeptics in self-isolation. And I think what we'll be doing is, this was a Merseyside Skeptics event, but next week there'll actually be a Skeptics in the Pub online event from everyone involved in Skeptics in the Pub, who will be putting on another talk next week. So stay tuned for more information about that. Normally, we would ask you for donations at the end of an MSS event. What we'll say this time is, instead of giving the money to us, try and give the money to something that's going to be help people. So maybe that'll be give it to a charity that's doing some work in that area. Maybe it's buying things for your local food bank, because there's certainly people who are quickly finding out that they don't have very much to live on and have got a little bit of time to figure out how to make that work. And anything we can do to make it easier for those people to keep going in the meantime, the better, really. I think we'll put more information about this up all online, and I think this talk will also be available as a video online after the fact as well. So we'll put out the details when that's available. Other than that, thank you so much, everyone, for coming. Thank you for being part of this first experiment. Thank you for all your excellent questions, and we'll see you next week, I think. Bye now.